Yeah, as Andrew said, my name is Rupert Cotter, and this summer I've been in the HIVE working on this project, Visualization of Human Brain Function Captured with Near-Infrared Spectroscopy. And as Andrew said, this was supervised by, primarily by Dr. Hanieh Bakhshayesh, and from within the HIVE here by Carley, Tillett, up the back. Yeah. So, quick little disclaimer before we get started on this. I'm an engineering student, and I'm in no way a health science student. So if you do notice anything a little bit questionable throughout this, please call me out on it, but just wait till after the presentation. Easy. So to get started, I think we should run down, do a little breakdown of what's coming up. So first of all, I want to take a look at why we're actually doing this project. What's the point? Then we're going to have a look at what actually is near-infrared spectroscopy. And then following that, we'll have a look at how the problem is solved. And then why this might be interesting into the future. So, right. First things first, I think it's good to break down this title because it is a little bit of a mouthful and have a look at what we're actually trying to do. We want to reconstruct images detailing human brain function and how it changes across the brain. That's what we're trying to do, and currently this task can be done. There's a few methods of doing that already. You might be familiar with things like fMRI or CT scan. And if you're familiar with those, I'm sure you're probably familiar with potentially how complicated they are, not to mention expensive, or even that they might come along with some drawbacks, whether that's powerful magnets in the case of MRI or the large radiation dose that goes along with a CT scan. Now, near-infrared spectroscopy, this is our device here. The devices are much smaller, more portable, and use much simpler technology, not to mention stylish on the forehead as well. But one thing I want to illustrate is how simple the technology that these use. So these are two examples of near-infrared devices. This is the one we've got, potentially for people online, and this is just a different model. How they actually work and what they actually are is something we probably need to talk about. So what is NIRS? I think it's best that we probably start breaking down the name first. Let's start by looking at spectroscopy, and then we'll look at light afterwards. So put incredibly simply, we can just say spectroscopy is an investigation of light's interaction with matter. But in our case, the light we're dealing with is near-infrared light. So what is near-infrared light? Something we come in contact with all the time, but on the surface it could sound complicated, like could be like strong magnets or radiation, but it really is something super simple. And to illustrate that, I have this. This is just an ordinary TV remote. And what these use to communicate with TVs around us is a little near-infrared LED in the front. Now I can press a button, and you shouldn't be able to see that anything's going on. But if I show you this video here, you can in fact see that the light does light up. Now that's the same thing as the LEDs on our headband. So there's 8 LEDs, and you can see when we start a recording, the lights light up. That's not something we can see with our eyes, but my phone does manage to capture near-infrared light. And the point of this is to illustrate that some of the technology that these use really is such a commonplace, ordinary thing. And we use it every day to do something as trivial as change the TV station. That being said, how does it work? How does it use this light to capture brain function? Essentially what it does is it uses the fact that the body is incredibly transparent, or reasonably transparent to the specific wavelengths of light that this device uses. And I say wavelengths here because it does use 2. Each one of these being tuned to be absorbed by either oxygenated or deoxygenated hemoglobin. So that allows us to track the changes of oxygenation within the blood, which of course changes as a result of mental workload. So we can actually take a look into brain activity through the lens of oxygen concentration using one of these devices. Cool. So what is the problem that we wanted to solve? We essentially wanted to reconstruct images of how brain activity changes across the brain. And there's a little bit of a process that I use to go about doing that. We can break it down into roughly these 4 steps. The first of which, we had to develop a test that we could then go and gather some meaningful data with. And we can then take that data through a processing stage, which we can break up into 2 steps itself. The first being some pre-processing, and the second, what we're after, visualization. Rightio. So taking a look at the first one of these, the main constraining factor for which test we had to pick simply comes down to where the device sits. And it sits on the forehead, so just above the prefrontal cortex. So to pick a test, we essentially had to decide on a test that would evoke a response from the prefrontal cortex, which is good. It's a nice thing to be constrained by. So what we ended up landing on was, oh, don't mind me, I've forgotten to take a little note for myself out. Yeah. So what we ended up landing on is an NBAC test. An NBAC test is a working memory test. Essentially what you have to do is, what the participants had to do was be presented with a letter on screen at a time, and you had to verify whether that letter was or wasn't a match with the previous letter. And in the case of the test we're going to look through, you have to compare it to the letter that came just before it. To get a better understanding of this, though, I think it's good we do a little practice run together. So I'm actually going to get all of you involved. You're going to have to call out yes for a match and no for not a match. We'll go through and we'll do 4 letters as a test first, and then you're going to have a shot at the proper test in a second. So that's our first letter. We want to compare the next letter against this. So let me hear you. Oh, music. Brilliant. And again? Go. Hey, what do you know? It even says match. Brilliant. That's it. So that's what we've got to do. In the actual test, though, you're only going to see 1 letter at a time. So you're going to have to remember the ones that came before it. So just like that, everybody nice and loud, we're going to start a test. So take it away.
Yes.
Nice. Not bad. Fairly straightforward, hey? Not too bad. So that's what we call 1 back. The test we did actually had 2 harder levels than that, which was 2 back and 3 back. And you might be able to guess, based off the name, that in 2 back, you're not guessing off checking the previous letter, you're checking 2 letters prior. And 3 back, 3 letters prior. So it gets pretty tricky. You can ask anyone that's done the test that it is definitely a test, that's for sure. Yeah, so what we've gone and done there, we've conducted the experiment, we can then bring it through to processing. And the first stage of this processing is to come and take our data and start cleaning it up. Basically because the device is quite sensitive. It's particularly sensitive enough to pick up things like movement, whether that's movement of the body, or more particularly movement in the eyebrows being quite intrusive, or even things like the breath or heartbeat in the signal. So what I've got here for you is just a small snippet of a signal. And in the red graph, in the red trace, we can actually see this oscillation up and down, and that's the presence of a heartbeat. Although this itself does tell us one useful bit of information, that my subject was in fact alive during the test, which is good. Yeah, anyways, so what we can do is we can see that this is again just a snapshot of some raw data on the left, full of noise, heartbeats, bumps up and down, and we can take it through a few stages of processing, of filtering and averaging, to end up with a much cleaner, friendlier looking signal like this on the right. And we can do that for all of our signals across all our participants, working towards a goal of extracting what we call a HRF, or hemodynamic response function. What this HRF is essentially a trace that contains the information of, it contains the behavioral response that someone's brain exhibits when presented with a stimuli, whether that's the onset of starting a task, like the little task I got you to do, or in this case, one person who's just participating in a rest period. This function is important because this is actually what we want to plot over the brain. We want to see what someone's response is when they start a task. So that can take us through to our final end goal. What we're working towards here is visualization. I'll bring those graphs back up in a moment. Actually not, I'll bring them up now. Let's go. So what we've got here, this is one snippet of a visualization. I've got the same response function here plotted. This is one of our subjects during a rest period, and this is just a 2-second snapshot into the response. And you can see in the graph on the left, it's the time period it is an example of is in the little square dotted box there. but I've made a video for this. So we can actually see as time goes on how the response changes on the brain against the graph. So take a look at this. There we go. So what we can see a couple of things here. We can see the activity changing around the brain, and we can also see that due to the warmer colours, that this subject was experiencing a decrease in oxygen. The cooler colours, sorry, this subject experienced a decrease in oxygenation over that test. Right, but this is all well and good. We've got one subject looking at one rest period, but what other things can we do? Perhaps another thing we could compare two different periods. So in this next slide, what I'm going to have on your right side up, no, your left, I guess, up here is a different subject undertaking a rest period, and on the right, they're going to be undertaking a test period. So we can compare the two videos side by side and see what oxygenation looks like during a test and during a rest. So, yeah, so that's it. So that's just a little 20-second snapshot. I've sped it up a couple of times for this. But what we can see is a few things. We see the warmer colors here during the active, during the test response. And that's evidencing the fact that there's an oxygenation increase over when we were looking at the rest period on the left. It's kind of handy. It allows us to do a few things. We can, without necessarily diving into looking at too many numbers and things, we can, once we've developed this system, we can look at brain activity on the surface level like this, just at quite a low level, which I think is really good. It's nice to be able to see things sometimes. But that begs the question. Why is this exciting for the future? What's the point of all this? So, there's a few good possibilities that could come, but let's look at the immediate future first. One potential thing we could do is integrate this method of visualization of data, looking at data that we've recorded in the past. We could integrate that with a live stream of data, allowing us to recreate these images as they occur, giving us a look into someone's mental activity as it's happening, which could be potentially useful. And looking into the future even further, that could have all sorts of applications. One could be, say, the development of a system looking at fatigue. So we could generate a system that analyzes someone's mental alertness, and it could potentially even give a warning to someone when their alertness drops below a certain level. Another thing we could do is create an interface between a computer and a brain. So we could set up a system that allows a brain to solely control some kind of device, not having to, just using this mental activity. One project I've seen is starting this year is an honors student is looking at creating a NIRS brain activity controlled wheelchair. So these sorts of things have lots of different applications, whether it's more practical or more practical in the sense of doing something. Right, so gone through a few things there, but hopefully you can kind of see that with a little bit of setup work, we can actually take a snapshot of what someone's mental activity looks like. And with a bit of processing, we can actually overlay this on the brain and generate a nice image to look like. So you can see a nice image to look, so you can see what the activity looks like. But one thing I just want to leave you with is that if you think any of this sort of NIRS stuff is quite interesting, I think there's a lot of stuff going on in the future potentially, so you might want to just watch this space. Thank you very much. If I could just spend one quick moment acknowledging everyone. So first of all, thank you to Dr. Hanieh Bakhshayesh for her help and guidance throughout this project, and of course, Dr. Darren, Professor Iain, and Ravindu for little bits of help and insight along the way. Of course, the Faculty of Science and Engineering for making this possible, and definitely everyone within the HIVE here. Wes, Carley, and Andrew, as well as all the other interns, have all been terrifically helpful. So thank you very much. Cheers.